The efficacy of pharmacological growth hormone. Because GF has so many roles in metabolism, including growth-promoting actions in tissues, the pharmacological use of GH has unknown and unpredictable results, especially in people with normal pituitary glands. It appears that the role of GH in muscle tissue is not related to events involving immature muscle fibers, since it has few direct effects on embryonic muscle tissue cultures. The enhancement of the contractile unit by GH interaction would appear to contribute to the development of the intact muscle, help in metabolic aspects, and affect subsequent force production characteristics. However, further research is needed to clarify exactly how GH is involved with exercise-induced hypertrophy. A review by Rogel showed that GH treatment alone is not effective in causing the strength increases, and that involvement of the total motor unit is probably necessary. Although size increases are possible, the quality of the muscle may be compromised owing to limitations in activation and control. Any apparent ergogenic effects may be outweighed by a wide variety of secondary effects not related to strength or size changes in muscle tissue. It seems plausible that the endogenous mechanisms related to the exercise stimulus have greater specificity and are more effective than GH injections for muscle strength and hypertrophy development. In fact, exercise-induced hypertrophy is apparent quite different from hypertrophy results from injections of GH. The force production of muscle fibers consequent to exercise-induced hypertrophy is superior. Furthermore, biological timing of cellular events in muscles and releases of GH may be crucial for optimal interactions between the exercise-stressed muscle and GH, with the many molecular weight variants involved in the complex picture of GH regulation and interaction. Simplistic views of pituitary function are outdated. Growth hormone is important for the normal development of a child and appears to play a vital role in adapting to the stress of resistance training. However, GH injections result in a wide variety of secondary effects not related to changes in muscle size or strength, and can in fact result in hypertrophy with less force production than results from exercise-induced hypertrophy. Growth hormone responses to stress. Pituitary hormones, propionmelanocortin, PQMC, GH, and prolactin respond to a variety of exercise stressors, including resistance exercise. GH contractions increase and respond to breathing, holding, and hyperventilation alone, as well as to hypoxia. It appears that the significant stimulus for 22 kDa GH release is increased hydrogen ion and lactate concentrations. Not all resistance exercise protocols demonstrate increases serum and GH contraction. Van Helder et al. Observed that when a light load, 28 of 7 RM, was used with a high number of repetitions in each set, no changes in the serum concentration of GH occurred. That's 28% of 7 RM. In resistance exercise, an intensity threshold probably exists when longer rest periods, greater than 3 minutes, are used to elicit a significant stimulatory response of GH to resistance exercise. This may be due to the metabolic connection with the glycolytic metabolism, at least for the 22 kDa variant. Depending on the load, rest, and volume of exercise of the resistance exercise protocol, different GH responses occur. In a study designed to sort out the different variations related to GH increases, Kramer et al. Found serum increases in GH are differentially sensitive to the volume of exercise, the amount of rest between sets, less rest, higher GH, and resistance used 10 RM produces higher lactate values and higher GH response. When the intensity used was 10 RM, heavy resistance, and 3 sets of each exercise, high load work approximately 60,000 joules, combined with short 1-minute rest periods, large increases were observed in serum GH concentrations. The most dramatic increases occurred in response to a 1-minute rest period when the duration of the exercise was longer, 10 RM versus 5 RM. Because such differences are related to the configuration of the exercise session, rest period length, it appears that greater attention needs to be given to program design variables when evaluating physiological adaptations to resistance training. A recent study by McCall et al. Linked GH responses and muscle changes with resistance training. GH appears to be related to the changes in muscle with training, but it is unclear how the various types of GH are involved in this process. The lack of high correlated relationship in this study may indicate a need to account for more than one type of GH. Excuse me. Growth hormone response in women. Throughout the menstrual cycle, women have higher blood levels of GH than men due to higher frequency and amplitude of secretion. Hormone concentrations and hormone response to exercise vary with menstrual phase. Although the mechanisms of this variation are unclear, Kramer et al. Found that during the early follicular phase of the menstrual cycle, women had significantly higher GH concentrations at rest compared with men. Furthermore, when using a heavy resistance exercise protocol characterized by long rest periods, 3 minutes, and heavy loads, 5 RM, GH levels did not increase above resting concentrations. However, when a short rest, 1 minute, and moderate resistance, 10 RM, resistance exercise protocol was used, significant increases in serum GH levels were observed. This suggests that hormonal response patterns to different resistance exercise routines may vary over the course of the menstrual cycle, owing to alterations in resting levels. It is interesting that acute aerobic endurance exercise shows higher post-exercise concentrations in women than men, but no difference has been seen in the genders in response to acute resistance exercise. The effects of periodizing resistance training over the course of a menstrual cycle remain to be examined, and more research is needed to elucidate any sex-related neuroendocrine adaptational mechanism. At present, women's reduced concentrations of testosterone and different resting hormone concentrations over the course of a menstrual cycle appear to be their most striking neuroendocrine differences from men. How such differences relate to training adaptations, the development of muscle tissue, and expression of strength and power remain to be demonstrated. Training adaptations of growth hormone. It appears that GH levels need to be measured over longer periods to see whether changes occur with resistance exercise. The area under the time curve, which indicates an array of pulsatile effects, tells whether changes in release have occurred. The response of GH to resistance training have not been extensively studied, but observations of normal single measurement of resting GH concentrations in elite lifters suggest little change. It is likely that differences in feedback mechanism, changes in receptor sensitivities, insulin-like growth factor potentiation, diurnal variations, and maximal exercise concentrations may mediate GH adaptations with resistance training. The typical trend for training-related changes in GH appear to be reduction in GH response to the absolute exercise stress and alterations in GH pulsatility. Characteristics. The reduction of the 22 kDa GH exercise-induced response with training perhaps indicate potential interactions with other molecular weight forms. Individual responses over a 9-month period were highly variable, with no significant group changes over time in a group of elite weightlifters. Those are unpublished observations. We are far from understanding how GH changes with long-term resistance training in athletes. Insulin-like growth factors. Some of the effects of growth hormone are mediated through small polypeptides called insulin-like growth factors, IGFs, or somatomedins. IGF-1 is a 70 amino acid polypeptide, and IGF-2 is a 67 amino acid polypeptide. The function of the latter is less clear. The liver secretes IGF after growth hormone stimulated in liver cells DNA to synthesize them, a process that takes about 8 to 29 hours. Typical of many polypeptide hormones, both growth factors are synthesized as larger precursors molecules. which then undergo processing to form hormones themselves. IGF travels in the blood, attached to binding proteins, of which there are at least 6 types, are released as free hormones to interact with receptors, and are regulated by various factors. Blood levels of IGF are usually measured as either total levels bound in free or free IGF concentrations. Circulating IGFs are associated with binding proteins. At least 6 circulating binding proteins have been identified that regulate the amount of IGF available in the plasma. IGF-1 binding protein 1 to 6 with BP1 and BP3, and most studied in response to exercise. Each responds to exercise stress independently and has its own biological actions. Binding proteins are important factors in the transport and physiological mechanisms of IGF. IGF has been shown to stimulate the secretion of its own binding proteins from within the muscle cell itself, thus modulating the cell's responsiveness to IGF. The circulating IGF binding proteins play an important role in restricting access to the IGF peptides and receptors, and are influenced by growth hormone concentration. Other factors such as nutritional status and insulin levels have also been shown to be important signaling mechanisms for IGF release. The nutritional influence of IGF transport, production, and regulation controls is a dramatic variable affecting its cellular interactions. Acute changes in nitrogen balance and protein intake and nutritional status affect a variety of mechanisms. It also appears that binding proteins act as a reservoir of IGF and release from the binding protein signaling available of a receptor on the cell. This allows IGF to be viable for a longer period of time and can theoretically reduce the amount of degradation on IGF. In strength training, many of these mechanisms are influenced by the exercise stress acute hormonal response and the need for muscle, nerve, and bone tissue remodeling at the cellular level. The dramatic interactions of multiple hormones and receptors provide powerful adaptation mechanisms in response to resistance training and can contribute to the subsequent changes in muscular strength and size.